Friends, we were discussing about the classification of HTS methods or assays on the basis of the format, and we discussed that we can broadly categorize them into in vitro assays and in vivo assays. Now, in vitro assays, we have biochemical assays, we have cell based assays, we have binding based assays and phenotypic assays. So, if we talk about biochemical assays, in the endpoints of such assays are largely determined by the fluorescence that is produced, by the absorbance or by the luminescence. And the techniques that are used in these assays are fluorescence polarization, scintillation proximity assay, FIDA, FRET, TR FRET, alpha screen, alpha LISA and so on and so forth. Then we have cell based assays wherein the impact of the test drug is observed on the cells. In such assays, the detection methods include the imaging of a cell, the fluorescence that is emitted by the product or the luminescence that is produced by the product, drug receptor product or drug protein or drug enzyme product. Then various techniques that are used to screen the drugs using cell based assays are reporter gene assays, then high content imaging we have discussed, then automated patch clamp, fluorescent image plate readers, TR FRET, atomic absorption spectrophotometry and alpha screen assays. If we talk about binding based assays which largely deals with the binding of drug molecule to the receptor, protein binding, protein nucleic acid binding and so on and so forth. So, we can say that these type of binding assays help in measuring the direct molecular interactions. And again we can detect such kind of interactions by fluorescence or electrochemiluminescence. And the techniques that are used are mostly overlapping for fluorescence polarization, TR FRET, alpha screen and so on and so forth. And last is the phenotypic assays, and we have discussed that these largely deal with the morphological characters, impact of the drug on the morphological characters. The second category is of in vivo assays, wherein the impact of drug is observed inside the entire organism. And largely two organisms are most widely used in high throughput screening: Caenorhabditis elegans and other one is the zebrafish. So, C. elegans is nothing but it is a nematode, and zebrafish, as the name indicates, it is a fish. The advantage why these are widely used, because we have fully elucidated the genome of these two organisms and we can easily see the impact of the drug on these two. Further, zebrafish specifically have a very fast growth rate, and moreover its larva or embryo are completely transparent. That also helps in making the analysis or screening or studying the impact more easy. Now, if we compare the biochemical and cell based assays in high throughput screening, as we discussed that biochemical assay largely used a purified target, and these purified targets can be an enzyme, it can be a protein, it can be a receptor, and therefore such kind of assays allows us to have a very precise and direct measurement of the compound interaction with these specific targets. But the problem with the biochemical assays is that they do not provide the impact of drug under physiological environment, because these are being carried out outside the cell. Whereas in case of cell based assay, the drug, the impact of drug is studied under the physiological environment, wherein various other interactions like cell matrix interactions are there, cell membrane and drug interactions are there, that can be studied and the impact can also be analyzed. Then since these cell based assays are carried out inside the cell, then they provide us the complex signaling. And why we call them as complex signaling? Because when the drug enters the cells, then it has so many interactions. Normal physiological processes are going on inside the cell, and then we get the such kind of complex interactions. But these interactions are invaluable for understanding how compounds basically affect the complex cellular processes. Then biochemical assays are simple and less biological complex, because we have isolated the purified target and we are just observing its interaction with the test drug. So, no other physiological process is interfering in this interaction. So, these are simple and less biologically complex, and therefore, such kind of biochemical assays are an excellent tool for initial screening of the drug molecules. Whereas in case of cell based assay, we largely use them to confirm the results of the biochemical assays, because no doubt the drug has shown a very good interaction in a biochemical assay outside the cell. But as we know, cell is having its own complex interactions going on. So, how that drug will behave in such environment. So, in order to confirm the results of biochemical assay, cell based assays are widely used. But the downside of cell based assay is that they do not measure the direct target engagement. So, we do not know which kind of interactions are taking place with the proteins or with the membranes by the test drugs. Now, this is just a crux of the entire classification of high throughput screening assays. As you can see that we can broadly categorize them into in vivo assays, which largely involves the usage of zebrafish, for example. Then we have in vitro assays, which can be broadly classified into cell based assays and biochemical assays. Again, we know that cell based assays are largely deal with the effect of drug on proliferation, the cell viability, protein interactions, and so on and so forth. And biochemical assays are further classified into enzyme assays, where we observe the impact of drug on an enzyme molecule, whether it upregulates the enzyme or it inhibits the enzyme. Then we also see the interaction of the test drug molecules with various kinds of receptors and effect on the hormonal secretions. Now, we will discuss some of the techniques that are used in high throughput screening, and I have further categorized these techniques into fluorescence intensity based assay techniques. Now, as the name indicates, fluorescence intensity, here we measure the intensity of the fluorescence that is produced as a signal, and amongst these, fluorescence polarization is one such assay. Now, in fluorescence polarization, what we do is we tag a drug molecule with various dyes, like fluorescein is there, rhodamine is there, cyanine is there, or naphthalimide dyes. Now, once the drug molecule is bind with these dyes and a polarized light is passed through it, since the drug molecule is very small, it will show a rapid rotation, which will change the polarized light which is passing into a depolarized light. And once the light is depolarized, we get no signal. On the other hand, if this drug molecule binds with a receptor or a protein or an enzyme, the entire size of this combination increases, and when we pass the polarized light, the speed of rotation decreases drastically, and the outcome is also in the form of polarized light, and this is used as a signal which indicates our drug molecule has bound to the receptor. Then second technique is the bacterial surface layer assay, which are commonly known as BSLAs methods. Now, these are largely used for screening the antibacterial agents. What we do is we use the surface layer of the bacteria, which is commonly known as S layers, as the biological sensor. We convert these surface layers of bacteria into the biological sensors by tagging them with a fluorescent compound. Now, a fluorescent compound could be such that when it is attached to a viable bacterial cell, it will emit fluorescence, or it could be a fluorescent compound which will not emit fluorescence when it is tagged to a viable bacterial cell, but when it is free from that membrane or the surface layer, it will emit the fluorescence. So, if a drug is having antibacterial action, it will disrupt the bacterial membrane or damage the bacteria or will kill the bacteria. So, if that fluorescent molecule is such that it will give fluorescence only with viable bacteria, So that signal will be lost if your drug will kill that bacteria. On the other hand, if the fluorescent molecule that we have used is such that it will only fluoresce when it gets detached from the bacterial surface, then if your drug is having antibacterial action and it damages the bacterial layer, the fluorescent tag will get free and it will emit the fluorescence. That will also give you an indication that your test compound has an antibacterial activity. The second category is of fluorescence proximity based assays. Now, as we discussed that the previous assays were dependent upon the intensity of the fluorescence was produced. Here we are talking about the proximity based assays. So, the name indicates that these assays are largely dependent upon the distance between the two molecules of interest. And it has been found that for such assay, the most widely used is fluorescence resonance energy transfer, FRET, or time resolved fluorescence resonance energy transfer, that is TR FRET. Now, in such assays, the distance between two molecules should be 1 to 2 nanometer, only then this assay will become active. And this much small distance only occurs when the two molecules interact with each other. So, what we do is on one molecule, say drug molecule, we attach a fluorescein molecule which emits fluorescence which is very short lived, and on the other molecule or the now this fluorescein molecule act as a acceptor molecule, and on the another protein we bind a donor molecule. So, when we pass a light at 680 or any specific wavelength, the donor molecule will absorb the light and it will donate the energy. So, if the two protein molecules are close enough, this energy will be accepted by the acceptor molecule, and once the acceptor molecule accepts this energy or absorbs this energy, it will start emitting fluorescence. And this is how we come to know, yes, there is an interaction between the two proteins. Now, if there is no interaction and these two protein molecules are far away from each other, that is the distance between the two is more than 10 nanometer, then there is no transfer of energy will take place from the donor to the acceptor and we will not get the fluorescence. Now, as I told you that fluorescein molecule was widely used, but the problem with it, the fluorescence that was emitted was very short lived, in the range of nanoseconds, and because of that there was a high background fluorescence in the assay which interfered with the actual results, and therefore gradually this fluorescent molecule was replaced by lanthanide fluorophores, which largely include europium and terbium cryptates, which have a fluorescence shelf life or a fluorescence life of milliseconds in comparison to the nanoseconds of fluorescein. So, what happens is when we use lanthanide fluorophores, so when there is an interaction, the background fluorescence, which is very short lived for nanoseconds, it will come and it will not be recorded, whereas the fluorescence of the lanthanide fluorophores, which is long lived in milliseconds, that will be coming after that nanosecond fluorescence has been finished, and now the background fluorescence drastically reduced, which improved the overall results of these assays. And when we use lanthanide fluorophores, it becomes a time resolved FRET assay. Second one is the alpha screen, that is amplified luminescence proximity homogeneous assay, and as you can see that for this assay the distance between the two molecules of interest should not be more than 200 nanometer. Now, what we do is we have two micro beads in this, and one is acceptor bead, other one is the donor bead. Now, we take the drug molecule and we take the target protein molecule or the receptor molecule. Now, the drug molecule can be bound to either by the donor bead or the acceptor bead, and the target protein is then bound again, it can also be bound to either acceptor or the donor bead. So, what we do is, for example, we take the test drug and bind it with the donor bead, and the target receptor we bind it with the acceptor bead. So, if the mixture is then exposed to a light having a wavelength of 580 nanometers, 480 nanometers, and when these two molecules come close to each other, then what happens is, as the light is absorbed by the donor bead, it will start losing its energy, and that will convert oxygen into a free oxygen radical, and this free radical is then accepted by the donor bead on which is present on the protein of interest, and it will start emitting light which will be having a wavelength range of 520 to 620 nanometers. So once this type of light is emitted, that indicates there is an interaction between the drug molecule and the protein molecule. But the problem with alpha screen assay is the broad range of the wavelength of the light that is emitted, that is 520 nanometers to 620 nanometers. So in order to further improve this assay, alpha LISA assay was developed, wherein the anthracene or rubine pigments which were used in case of alpha screen assay were replaced by the europium chelates, which have a specific emission wavelength of 615 nanometers. Rest of the principle was same as that of alpha screen assay. So what happens by replacing anthracene or rubine with europium chelates? You can see the broad graph was of the alpha screen assay. The screen or the graph ranges from 520 to 620 nanometers, whereas in case of alpha screen assay, we get a very sharp and a narrow peak of the signal at only 615 nanometer, which significantly improve the sensitivity of the test, which significantly improve the overall outcome of the assay. These are few of the advantages of alpha LISA. As we discussed, it was more sensitive than alpha screen assay, and it has a very robust performance with the reduced assay times. Further, it was easily adaptable to the automated systems of the HTS. Now we will discuss about a cell based assay which we normally which is most widely used, that is the reporter gene assay. So basically, if we look into the definition of reporter gene, so it is a genetic sequence which is linked to the regulatory element of the gene of interest. Now if we talk about the regulatory element of the gene, which is normally called as the promoter or enhancer of the gene. So what we do is, we take the reporter gene, which is a specific genetic sequence, and link this sequence to the promoter region of the gene of interest. So if our gene of interest, the expression of that gene increases, the reporter gene will also get active. So we will discuss this with an example. So we bind it with the regulatory element of the gene of interest, such as promoter or enhancer, which helps in tracking its activity. So when the regulatory element is active, the reporter gene is expressed, producing a detectable signal like fluorescence, luminescence, or a change in color. There are three most widely used protein molecules in reporter gene assays, like green fluorescent protein, luciferase gene, and beta galactosidase gene. Amongst these three, luciferase gene is most widely used. Now luciferase gene is a gene which helps in increasing the expression of the luciferase enzyme, which is found in fireflies. Now this luciferase causes the breakdown of luciferin protein, which helps in emission of light, and that is why fireflies glow during the night time. And this whole process we call it as bioluminescence process. So what we do in these assays, we take luciferase gene as the reporter gene and we bind it to the promoter region of the our gene of interest. Then we expose or we incubate this mixture with our test drug molecule. So if our test drug molecule has an positive impact on the gene of interest, what will happen? The promoter region will get activated. So as soon as the promoter region of the gene of interest gets activated, it will activate the reporter gene as well, which is luciferase gene in our case. So as soon as this reporter gene gets activated, it will start producing the luciferase enzyme, which will act on the luciferin protein present in the SA mixture, and it will degrade this luciferin protein and a light will be emitted. And this light is captured as a signal. So this was just one example how the reporter gene assays work. But there are certain downsides or limitations of reporter gene assay. Now these reporter genes can sometimes interfere with the endogenous functions of the gene of interest, and which eventually has a negative impact on the overall cell behavior or its survival, because these reporter genes are nothing but these are the exogenous DNA molecules, and we are inserting these exogenous DNA molecules into along with our gene of interest, which can sometimes interfere with the functioning. These reporter genes also sometimes they induce the silencer activity in the gene of interest. Now, silencer activity again is nothing but a sequence in the gene which basically prevents the expression of the gene, or which may influence the promoter region of the gene, and therefore the activity of the gene reduces. So sometimes these reporter genes also activate the silencer activity, which eventually downregulate the gene expression, and we may get false negative test. Though the drug has a positive impact on the gene expression, but because reporter gene has activated the silencer activity, this may give us a false negative result. Then these reporter genes are impacted by the cellular and biological environments, which can alter the activity, leading to the variability in the results. So these were few limitations of the reporter gene assay. Another one is the high content analysis or high content screening or high imaging assay. So this image give you a brief flow chart of the events that normally occur during this assay. So first one is we select the suitable cells on which we want to study the effect of the drug. Then these cells are seeded onto a suitable microplate of appropriate density and appropriate number of wells or the size of the wells, and then all other reagents, test drugs or other assay reagents are automatically added by the automated instruments, and they are allowed to for specified period of time to stay or to incubate under specific temperature and humidity conditions. So once the incubation time is over, the automated image capturing will occur. So the equipment will capture image of all the wells that are there on the microplate. Say for example, you are using a 96 well plate, then image capturer will capture image of all the 96 wells, and these images are then analyzed with the help of software. Then we generate the data, and then this data is interpreted. Now in this whole process, if we talk about, then there are few critical steps. First one is the selection of the microplate, which is considered one of the most critical steps in high content analysis or imaging cell assays. Why? Because the thickness of the walls of the well, the base of the well in the plate, whether it is flat or curved, all such factors have an impact on the image quality that will be used for the data interpretation. No doubt the appropriate selection of the cell line or the cells should also be there, and all the temperature and the humidity condition during incubation should be properly maintained, but selection of microplate is one of the most critical factor. So how the selection of the plate impacts the result, this is shown in these pictures. So if we see the first image on the left side, you will see this is an image of a well that is captured in a 96 well plate. So you can clearly see that the image in the central portion of the well is quite sharp, but if we look at the edges of the plate, the image becomes slightly blur. Now this could be largely due to uneven thickness of the plate at the edges, and when such kind of image is then analyzed by the software or algorithm which we are using in the software, that will give you the varied results, because at some places it will capture the cells, it will count the cells, or at some places where the image is slightly blur, it will leave those cells. Similarly, thickness of the cells also have an impact on the image. So as you can see on image on the right, the upper images were taken when we used a microplate having a cell thickness or the bottom thickness of 0.7 millimeter, and on the lower side we have a 0.19 millimeter thickness of the well base. So you can clearly see that image at the highest magnification, that is 40x, is slightly blur when we are using more thickened base microplates in comparison to the thin plates. So these were few examples that how the selection, why the selection of the microplate is very critical. We have to be very careful. The wall should have uniform thickness, the base should have appropriate thickness. If you are using a flat base, no issues in clicking the image, but if you are using curved base microplates, then it is always preferred to click image from the central region of the plate, because again the thickness will vary in the curved region. And the lower image shows you the area that is covered, and when we are clicking an image at 40x maximum magnification, the amount of data that is generated is somewhat around 230 GBs. And the same image, if we click at say 20x, 20 times magnification, then the amount of data significantly reduced to 60 GB. So it's not that you have to click the image, you have to select the magnification appropriately, because the amount, larger the amount of data, more the time the software will take to analyze it. Now I will just briefly discuss the protocol or the steps that are that occur in setting the protocol of high throughput screening. First one is the selection of target, or you have to select whether you want to go for a target screening or a phenotypic screening. So depending upon your requirement, you have to first finalize. Then second is the assay development. Then you develop your assay for the desired results. It's not only about the development of assay, but again you have to optimize your assay. There are various parameters that are required, that require optimization in an assay. I will just give you a glimpse of that as well. And then once you optimize your assay, then we have to validate whether our assay is giving the correct results or not. We have to validate it as well. Once development, optimization and validation is done, then we implement the high throughput screening. Then we acquire the data, we store that data, and then we analyze. Once the data is analyzed, we used to get either hits or we get leads. Now we were talking about the optimization of the assay. So these are various parameters that should be optimized, like automation capability of your assay, whether your assay is amenable to automation or not, what kind of plate you want to use. You want a flat base or a curved base. The walls should have a uniform thickness in order to avoid the edge effect, which we discussed during the high content imaging. Then the optical properties of the 92 well or whatever microplate you are using, because majority of the assays you have seen that they either use fluorescence as a detection or electroluminescence or chemiluminescence as a detection. So the fluorescence that is produced from one well should not interfere the results of the other cell. So that is why optical properties become important. Density of the plate is another important factor. So depending upon the type of the data, depending upon the number of the samples you want to analyze, you have to appropriately select the density. The other parameters could be the pH of the reagent you want to use, the buffer, what kind of buffers you want to use, the order of addition of these chemicals is again an important. Then the concentration of your substrate, it could be an enzyme, it could be protein. The incubation times, again it's important, for how much long you have to incubate the mixture of the test drug and the protein molecule. Then the drug concentration is another important factor which you have to optimize. So friends, this was about just the methods that are used in assays. Obviously we have discussed only some of the methods. There are so many methods, and discussing all the methods would be beyond the scope of this lecture. So in the next part of the lecture, we are going to discuss how this HTS has an impact in the field of natural products. Thank you.